Hi everyone, good morning and welcome to this joint webinar between Promega and Arctoris. My name is Hilly Pollard and I'm the product manager at Promega. Promega's Lumit SARS-CoV-2 Spike RBD ACE2 Immunoassay is the subject of today's webinar, and we have two presentations for you. In the first, Hicham Zegzouti from Promega's R&D team will introduce Lumit's immunoassays and how they work, with a focus on the SARS-CoV-2 assay. In the second presentation, Dan Thomas from Arctoris will discuss their onboarding of the assay and initial data. After the presentations, we will have a live Q&A session with a team of experts from both Promega and Arctoris. So please submit your questions in the Q&A box at any time during the talks, and we'll get to them at the end. Following on from Dan's talk, we've also put up a variety of resources along with a short survey, which we'd greatly appreciate you completing. I'd like to now introduce the first presentation. As Hicham is based in the US, this is a pre-recorded presentation.
Thank you for the introduction. Hello everyone. Thank you for attending this webinar with us today. My name is Hicham, and I'm a senior research scientist in the assay design division of Promega R&D. Today, I'm going to talk to you about the new addition in terms of assay development to our team. Today I will be talking about the Lumit SARS-CoV-2 Spike RBD human ACE2 immunoassay, and I will describe to you first the Lumit technology before I dive into the SARS-CoV-2 application. What we are talking about today is the SARS-CoV-2 and the COVID-19 disease. The SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2, and it belongs to a family of coronaviruses, which are large, enveloped RNA viruses. This respiratory disease caused by the SARS-CoV-2 has been named as COVID-19 by the WHO. As you remember, on March 11, 2020, the WHO declared the COVID-19 a pandemic, the first that is sparked by a known coronavirus. Since then, research efforts are underway to create vaccines against the virus and identify therapeutics to treat the COVID-19 disease. A little bit of biology to introduce the virus and the human cell interaction. SARS-CoV-2 uses the human ACE2 as a receptor for entry into the target cell. As you can see from this picture, you have the spike protein, which is a trimer. This virus actually interacts with the angiotensin converting enzyme 2, which is a totally different receptor in the human cell that doesn't have anything to do with virus. It's just the virus actually hijacked that ACE2 receptor to enter the cell through many steps that includes endocytosis, etc., before the viral RNA is released and transcribed, translated to the viral machinery. Particularly, the virus uses the RBD domain of the spike protein, shown here, and as you can see from the sequence of this spike protein, it has S1 and S2 subunits, and RBD is a part of the subunit 1, S1. And that's what the virus is using to bind to the ACE2 receptor to enter the cell. Over this year, we saw a lot of publications studying this virus, and we know that antibodies that are produced against the RBD domain are capable of viral neutralization and are now targeted, of course, for treatment and vaccine development efforts. So many strategies exist to look for therapies to counteract the SARS-CoV-2 infection, but one of them focalize on the S1 or the spike and ACE2 interaction. So one strategy is to identify inhibitors that could be neutralizing antibodies, peptides, small molecules that could prevent these two proteins from coming together and preventing, of course, the virus from entry. Of course, an assay that will detect the inhibitors of SARS-CoV-2 Spike RBD and the human ACE2 interaction would be ideal. So before we talk about the assay that we developed in my team, the current methodologies to detect inhibition of viral infection include plaque reduction neutralization test, which is a gold standard. It's called PRNT. This assay requires the use of a live virus and high biosafety level laboratory, BSL-3 level. So the way the assay works is you take a serum from infected patients that you want to see if it has a neutralizing antibody that can inhibit the wild-type virus. You mix them together and then you incubate them with Vero cell, E6 cells, to see if they will have the infection effect or not. And over different treatments and incubations, you look at plaque formation on a gel, where you image actually plaques, meaning if you have plaques that are formed, the virus was still active, and it means it wasn't neutralized. It means the serum didn't contain enough neutralizing antibodies. And if you don't have any plaques, it means the virus was neutralized. The second type of assay is similar, but it uses it's a surrogate assay that uses actually viral particles that are engineered to contain luciferase as a reporter, and also a spike protein that will allow that virus to enter the cell. It's not an infectious virus, but it can enter the cell through the S1 ACE2 interaction. This virus or this assay requires only a BSL-2 level, but it will require cell engineering, of course, to produce those viral particles with luciferase. The way it works is similar. You mix the viral particle with the serum that supposedly contain neutralizing antibodies, and then you incubate this with cells, human cells, and then you look for luciferase production or light production through luciferase. So these assays, both PRNT and VNT, are time consuming, and this is why there is a need for a fast and easy surrogate assay that can be used to detect inhibitors of viral entry or inhibitors of the interaction between the virus protein and the human protein. This is why we can develop an immunoassay that can detect the RBD ACE2 interaction. And in my group, we developed a Lumit Immunoassay for this interaction. But before I talk about the assay, what is Lumit? And before even Lumit, what is the engine of the Lumit assay? The engine of the Lumit assay is the nanolock binary technology, or nanobit. So in Promega, we have a small and bright luciferase called nanolock. This luciferase actually can be split into two pieces, one called large bit, which is 156 amino acid, and one is a peptide at the C terminal of 11 amino acids. If this nanobit luciferase emits light in the presence of furanumazine, but because when you split it, you have a very high KD between the two pieces, so it's a very low affinity interaction, they cannot emit light by themselves. So you need something like facilitated complementation. What I mean by that is you can attach them to two proteins that you think interact, or you can recombinantly produce them. And then when those two proteins interact, they bring the small bit and large bit in close proximity, and that generates light in presence of furanumazine. This has been published and used for many applications, But what we did is actually we took advantage of this nanobit interaction or this nanobit platform or technology to create the Lumit assay by creating antibody-antibody interaction or antibody-antibody detection of analytes. So the same way, if we conjugate a large bit to one antibody, small bit to another antibody, and in the presence of an analyte where the two antibodies will bind, the two pieces of nanobit luciferase will come together and they will create light. So how do we apply this to ELISA? So this is just a novel alternative to conventional immunoassay approaches. Here is just a reminder of the traditional ELISA workflow. Of course, as you can see, it has multiple steps involving several wash or incubation time-consuming steps. So what we did for Lumit, we actually took advantage of this nanobit technology that I showed you, and we started actually stripping different steps from the traditional ELISA because our assay is homogeneous. It means everything happens in the same way. We took the coating, there is no coating, everything happens in solution. We took the washes because everything happens again in solution with no coating, no blocking, etc. And we even went further. We took the steps of additions to include only one step of antibodies, either primary or secondary antibodies. This assay is easy and fast. It takes 30 minutes to 2 hours, depending on the application. It has a high sensitivity, as I will show you. You can detect a target in a low number of cells. It has a broad dynamic range, and it can be used in a different plate formats, 96 or 380 were 4 well plates. And it is, of course, homogeneous, so it means it's HTS compatible. So the three types of Lumit no-wash immunoassays consist of direct assay, where you have labeled primaries, and we have kits actually that can help you label your antibodies. This first concept requires primary antibody labeling. It maximizes the number of antibody pairs that you can test for your different targets. And we validated this assay for cytokines, peptide hormones, and metabolites such as interleukins, insulin, etc. Then we created also an indirect Lumit assay where instead of labeling the primary antibodies, we label the secondary antibodies. And now you are free to use any antibodies that are available to you as long as the two antibodies are from two different species. So we created an anti-mouse, an anti-rabbit, and an anti-goat with the small bit and large bit. It means you need to find two primary antibodies from two different species. This way, we created something that can be a universal approach where you can just plug and play. Anytime you change your target, you can get just two antibodies specific with a target and use the same Lumit secondary antibody. This way, you avoid labeling primaries. We validated this indirect assay that I will show you on intracellular protein targets and phosphorylation of specific signaling nodes. And of course, I will show you how we adapted this secondary antibodies to the PPI assay using RBD and ACE2. There is a third concept that uses Lumit. It's competitive binding, but we are not going to go through that. There are a lot of presentations that you can look at tracer binding, etc. So how about Lumit cytokine immunoassays? So the cytokine immunoassay, this is just an example of interferon gamma immunoassay to detect the cytokine release from cells. So first you plate your cells and treat them, and then add the antibodies because you are expecting the release of interferon, for example, for this application. You incubate for 60 minutes, you add the detection reagent, and you read. So the total assay time is 70 minutes. And you can create data like this. So this is a standard curve of human interferon gamma. You have very good sensitivity and broad dynamic range, and it mitigates actually the need for sample dilution, unlike ELISAs where sometimes you are outside the range, so you have to dilute your sample, for example, that you can see the level of cytokine, and it has an excellent cell-based performance. To show here is just a treatment of effector cells and target cells with a bispecific T cell engager titration that can actually trigger the release of interferon, and this is a cell-based, so you can see actually what is the effect of your antibody. We have multiple cytokine and metabolite assays in development, and for more information you can just ask your Promega representative, and soon probably you can see them on our website. Now, the one that we developed in my group, it's a Lumit Immunoassay cellular system, and we applied it to interrogate cellular signaling pathway regulation. Just as a matter of introduction, of course, you all know that protein phosphorylation is crucial in cell signaling, and in order to study pathways or signaling pathways, if you look at specific phosphorylation events that happen, like for example here in this NF-kappa B pathway, you have one protein that we can actually call it a node. This protein, which is I-kappa B alpha, after TNF activation of the pathway, it gets phosphorylated and degraded to release the P65, which is the real NF-kappa B, to go into the nucleus to transcribe genes involved in an immune inflammatory responses. Same way you have a STAT3, which is the cytokine pathway, JAK-STAT pathway. After IL-6 treatment of cells, JAK gets phosphorylated and activated, and it phosphorylated STAT3 or 5, etc. So those proteins that get phosphorylated can become a node in signaling pathways that you can analyze their phosphorylation to give you a report on how is the pathway activity. So the Lumit Immunoassay cellular system, it provides a rapid homogeneous assay system to quantify phosphorylation or even degradation, which is the total level of protein in those signaling pathways. How does it work? So let's take the I-kappa B alpha example. So here we have cells that you activate through whatever ligand or cytokine that will activate the kinase to phosphorylate its substrate, I-kappa B here. You add a lysis buffer, and everything is happening the same way here. So you have a plate, you have cells, you just add the lysis buffer, everything will be released in the well. And what we do, we add actually 4 antibodies here. So this is the indirect system that becomes actually universal. So we add 2 primary antibodies, one mouse anti-kappa B, and one rabbit anti-kappa B that bind to 2 different epitopes. And at the same time, you add the Lumit secondary antibodies. The one with large bit is an anti-mouse, and the one with small bits is an anti-rabbit. And it could be the other way around. It could be the anti-mouse with small bit and the anti-rabbit with large bit. Both combinations can work differentially. Some work better than others, but you will see actually, if you look at our website, we have some targets that use one combination and some targets use different combinations. So then when you add those to your cell, then you are looking, for example, at the total protein detection. Now both phospho targets and the non-phospho target are detected. Now the other way around, if you want to look at the phosphorylation of I-kappa B, you just switch one antibody. So instead of the rabbit anti-I-kappa B, you use an anti-phospho I-kappa B. And you add those again, and then you can see that now you are detecting only the phosphorylated target. And unlike Western, for example, where you see bands that the antibody is recognizing because of non-specific interaction, in Lumit assay, you cannot see that, because if you have only one antibody binding the target, you cannot generate light. So it's very specific. So it's a rapid add and read format to interrogate cell screening pathways with no medium removal, no transfers, and no washes. So this assay actually, when we developed it, we validated through looking at different number of cells. As you can see, we have a broad dynamic range. You can detect as low as thousands of cells. Here again is IxBa detection upon TNF treatment of the cells. So you can see there is a degradation here. So you have an increase in IxBa level with the number of cells. But when you have TNF, there is no IxBa. It means it's degraded. Same thing, we can treat the cells with MG132, and now you can look at the phosphorylation. So the phosphorylation increases with the TNF treatment the same way. This assay actually was validated in 96 well, and here is the protocol, and also in 384 with good sensitivity. So this assay was tested with multiple assays. Here is again IxBa with just 1 point. We treat with TNF, you see an increase of phosphorylation and decrease of total. And then when you treat with an inhibitor, you decrease the level of the activation of NF-κB, and you prevent degradation of IxBa using inhibitor. P65 is again another, it's the NF-κB protein. Here you can see that you can increase its phosphorylation, but of course you don't change the level of IxBa. To finish with the Lumit, this is the last application, which is when you look at drug discovery. So can you actually inhibit the pathways with different drugs? Here we looked at JAK inhibitors, compounds, so very good FDA-approved drugs that inhibit the JAK. So we are looking at phosphostat-3, so you can inhibit it with very low IC50s. And you can, of course, use antibodies that prevent the ligand IL-6 either to bind the receptor, or antibodies that will bind the receptor and prevent it from binding. And those also actually prevents the phosphorylation of STAT3. So for high throughput screening, you have a very good Z prime. So here we tested insulin activation of phospho AKT. And also we used an inhibitor that inhibits PI3 kinase that prevent this phosphorylation. So here we did like 140 wells with the same thing to calculate actually the Z prime for high throughput screening. High Z prime factor indicates that the Lumit Immunoassay cellular system is amenable to HTS application. So in our website actually, and this we validated multiple targets, phosphorylated targets, total level of those targets, and also we published the deciphering of these signaling pathways in this publication. Once you get this presentation in PDF, you can actually click on those blue things and it will take you actually to the application notes for this. So again, just to summarize, the Lumit Immunoassay platform is based on nanobit technology. It's a rapid, no wash, no transfer assay. It requires standard luminometer. It's scalable for high throughput use. And the platform includes ready-to-use kits as well as reagents to create novel detection assays, which is a labeling kit. And it's a universal format, and you can read about it in or listen to multiple webinars that we have that you can access in this blue link here to have more details of the different variants. So now I'm going to move for the last 8 or 10 minutes left to discuss about the new assay, which is related to SARS-CoV-2. Here, as I explained, we can use this Lumit technology to look at protein level, but we can use it also to look at PPI by using 2 antibodies on 2 different proteins. But here we used actually a different twist to it. So again, this is an assay that detects RBD from the virus and ACE2 from the human cell, to see if they can interact or we can disrupt their interaction using inhibitors. So We use an RBD that is tagged with a rabbit Fc domain from an antibody, and we use ACE2, which is tagged with a mouse Fc from an antibody. This way we can use our Lumit secondary antibodies. So why we choose to do it this way is we don't want to use antibodies that bind directly RBD and ACE2 in order actually the assay can prevent the binding of ACE2 and RBD or prevent the inhibitor from binding those. So we didn't want to interfere with the binding between RBD and ACE2. This way we chose to use the secondary antibodies that bind outside. The way it works is if you mix the two proteins and you add the two secondary antibodies, both antibodies will bind their cognate Fc, and then they will bring the nanolog subunits in proximity and create light. Very simple assay. This Lumit Spike RBD ACE2 is homogeneous also. It's add and read assay to screen for inhibitors. So before we go into that, so during development, we found that this interaction actually is very linear. You can produce light up to 2 nanomolar of those two proteins. And then, of course, here, if you want to use this assay for high throughput screen, you want to see after you generate light, how long this light is stable. This way you can do batch processing. You can do multiple plates if you want during HTS. So we found that the assay signal is stable for more than 100 minutes with approximately 75% remaining signal. And this way it's good for HTS. Now, to use this assay actually to look for inhibitors, you can use this kind of scheme. First, you add 5 microliter of either a sample that contains inhibitors, serum, plasma, neutralizing antibodies, anything that you suspect will inhibit. You add 10 microliter of the rabbit Fc RBD. You incubate for 30 minutes to allow any binding to happen if you want. And then you add the ACE2. You incubate for 60 minutes with the antibodies also, and you let everything to bind and to generate light. Once you finish 60 minutes, you adjust the detection reagent, and 30 minutes later, you record the luminescence. So it's an assay that detects this interaction with less than 2 hours. This assay can be used to detect these 3 types that I'm going to show you today. You have either small molecules or peptides, antibody libraries, or neutralizing antibodies from serum. We checked first if the assay can be miniaturized, so we tested it in 96 well compared to 384 well plate with low volumes, and you can see that the performance is the same, and actually it's an assay that is amenable to high throughput screen. We don't have any compounds that I can show you today that inhibit the RBD ACE2 because none exist yet. So hopefully people who will use this assay can find some, but we have data using antibodies. So the first we took just for a test to see if we can inhibit the interaction using 4 antibodies anti-spike protein. So 2 of them were really neutralizing and 2 of them were just very mild inhibition. And we also tested ACE2 in a competition assay with the ACE2 in the assay. So ACE2 recombinant his tag ACE2 was tested in a titration using the assay, and you can see we can inhibit the interaction between the RBD and the ACE2 of the assay. So this assay is amenable to testing antibodies. So we created a small antibody library by getting 18 antibodies that are known to either bind RBD or bind the S1 protein, or they are known to be neutralizing or not. So we just collected those and we created a little library to test. So here is the assay results. So we can see that many antibodies don't have a higher inhibition of the RBD ACE2 interaction, but some of them do, and really it can show you the different levels of inhibition. This is only one concentration. We took 13 of them and we did a titration to see the IC50 of inhibition. and 3 of them that really inhibit with high potency the assay. You can see them here in color, and all the others, they have a different degree of inhibition, but they have a very low IC50. So this assay can allow you to screen antibody libraries for antibodies, or for neutralizing antibodies that can inhibit the RBD ACE2 and hopefully the virus entry. So now talking about the virus entry, how can we use this assay as a surrogate for virus neutralization test? So I show you the PRNT in the beginning. It takes 4 to 6 days. Lumit assay, just by combining RBD ACE2 with the serum, you can actually do it in 2 hours. The same concept here, no need for BSL2 or BSL3, and you can run the assay as quick as possible with multitude of samples. So first, before jumping into serum samples, we wanted to see, by just using a serum from Sigma at 10%, how does it affect the performance of the assay itself. So we took 10%. We know that higher concentration of serum change a little bit the Lumit assay performance. It decreases the light. We want to see this specific one, how far we can go with the serum. We don't need to use higher serum when you do this kind of assays, but we want to start at 10. So at 10, you can see that you still have a good linear range of the detection. And we took this 10% and we spiked the one of the antibodies that we know is neutralizing, and we can actually detect with the similar IC50. The assay also is the signal is stable, so you can do high throughput screening for that if you want, or use multiple plates at once. But because we know that serum can have that inhibitory effect, we tested a multitude of negative serums, and we saw that each one has a different effect on the assay. So at 10%, probably we have that variability. So we want to see, can we diminish that variability by testing less percentage. So we took like 4 negative samples and 4 positive samples that you can see here. And we want to see if we go down to 5% or below, can we actually recover the light output and have less variability? And that's what we got. So 5% seems to work, or 5% and below, but we wanted to keep actually 5% serum as optimal condition, because this way we can have a decrease in the variability of negative serums and potentially detects positive serums with low neutralizing antibodies. Now we are ready to test actually the cohort of positive and negative PCR samples. So that's what we did. Here is the data. So first, to calculate actually the percent neutralization, we just look at control, just Lumit reagent as 0%, and PPI activity from you take the mean of pre-COVID-19 negative serums, and you make it as 100%, and then you subtract that from 100 to make the percent neutralization. And we decided to have a 30% inhibition cutoff, because that's way we can have all the negative serums below it to create 100% specificity. Then we look at the positive, and we see, like here in this example, our cohort of 40 samples, 32 of them were positives. So the 8 are negative for neutralizing antibodies. So it works really well. So we have also a collaborator in Brazil that have big cohorts, so 87 samples of COVID-19 positive and negative serum that were tested with the serology assay. So here you can see that by testing those serologically tested assays, they saw that, okay, our assay can detect the negatives and the positive in a specific fashion. And also you can also do some studies to look actually post symptom after COVID-19 infection. How does it go? And it's really, there is a good correlation there. The same thing, you can actually spread those post symptom or days post symptom, and you look at the positivity rate, and you can see actually that after 9 days you start seeing neutralizing antibodies. So this assay is really powerful for that too. So This assay is available in 2 different sizes, and it's available in our custom assay services. Kits are available anyway, and you can inquire about them talking to your Promega representative or looking at the website. I hope I showed you that this assay, the Lumit SARS-CoV-2 Spike RBD ACE2 Immunoassay, can detect different inhibitors of this interaction in a fast and easy way. It's amenable to miniaturization for high throughput screen, and also it can be used as a surrogate for a VNT to analyze patient serum NAB level. Thank you. And now I'm going to turn it to Danielle.
Hi, apologies for the slight technical hitch at the start of the talk. We hope you enjoyed it anyway. If you have any questions, please let us know in the Q&A box. We're going to go through these at the live session at the end. For our second presentation, I'd like to introduce Daniel Thomas, Head of Discovery Biology at Arctoris. Daniel's going to discuss onboarding of the Lumit SARS-CoV-2 Spike RBD ACE2 Immunoassay onto their onto their automated platform to generate clear, high-quality data. Over to you, Dan. Thank you.
Thank you very much, Hilary. First of all, I'd like to share with you a perspective that I first developed about 7 years ago, but I think it rings as true today, and particularly in the current climate, as it did back in 2013. So really, the challenge to our current paradigm is a growing pressure to deliver greater value with less resource is a familiar concept to all of us. And it's currently challenging our paradigm of drug discovery. The opportunities we have to profoundly impact discovery through the value of decision-making will be critical in defining our future successes as an industry. And I think this really encapsulates one of the reasons for my transition from large pharma, 2 decades in large pharma, into a small biotech company that is largely focused on data quality, reproducibility, and the delivery of premium quality data to clients around the world. So the strategic shift that we have seen over the last few years is really a confidence in the accuracy of research findings remains low. This is down to experimental reproducibility issues. There have been a number of high-profile article retractions. These result from inconsistent performance, poor validation of assays, inappropriate implementation or even absence of standard molecules, variability in the data and metadata quality with inconsistent capture, inconsistent tracking, and more importantly, subjective analysis, leading to a cost and failure rate cycle times that are all far too high. Now, there is a polarized opinion, or there are polarized opinions out there of automation and the externalization or outsourcing arena. But I think COVID-19 this year has really highlighted organizational operational vulnerabilities. It's prompted fundamental reassessments of strategic priorities. And this is where the Cloud Lab really offers its value. So Arctoris offers accelerated experimental onboarding, which I will come to at the end. It uses the highest quality reagents and instrumentation available. We eliminate almost all of the human variability that contributes to irreproducibility and variation in data. We can guarantee experimental reproducibility by coding production runs in computer code. We guarantee fidelity in the capture of all data and metadata, which allows us to deliver comprehensive electronic audit trails. And we can do this at a significantly reduced cycle time. We can turn assays, biochemical assays around in as little as a day. Cell-based assays typically slightly longer depending upon the duration of that assay. Now, I'm sure this is an image that most of you, if not all of you, will be familiar with around quality as viewed by precision and accuracy. So accuracy is how close to a measured value is to the actual or true value. This is something that is unique to biology. It is something that requires us to know something about our system in order to understand how close our measured value can get to that. Precision, however, is how close the measured values are to each other. And this is something that at Arctoris, through our ability to run directly comparable, reproducible assays, we're able to guarantee that precision to be at a much finer detail. We highlight outliers using modified Z-scores and statistical interquartile range analysis. We exclude outliers using median and median absolute deviation statistics in generating our robust readouts. And we ensure performance QC by being able to demonstrate the statistical separation between two unique values in providing a robust discriminatory difference. Now, variability comes in a multitude of sources. The quality of a measurement of data is crucial to robustly test any hypothesis. And what we need to do in setting up systems to run experiments is to ensure that we eliminate as much of those sources of variability as possible. Now, these can arise through measurement errors. So the validity of an assay is the ability of an instrument or assay to accurately measure what is intended. So this requires the inclusion of reference tests or standard molecules for comparison. Reliability is the consistency of performance of an instrument over time and between different observers, but I will come on to that in a moment. Repeatability is the test and retest reliability. So this is the consistency of measurements by a single observer over time. Now, random or chance errors result from not taking the same measurement in exactly the same way. Often these are ones that can't be predicted and may ultimately stay as a source of variance that we need to accept. Systematic errors, these are more consistent, they're reproducible and usually in the same direction, and they're often due to problems which persist throughout an entire experiment. Sampling errors, where different samples produce different results due to chance. This can result in false positives and false negative classifications. The false negatives are the ones that are probably most worrying. If we have a triage system in place, often false positives can be weeded out, but not finding the molecule in the first place may lead us down the wrong path from day one. Sample variability. Now, we have natural variability in reagents, particularly with cells due to physiological differences, batch-to-batch variabilities, longitudinal stability, potentially passage, that will introduce variability into that cell line. So we need to be aware of these. Observer variation, so we have intra-observer, so this is inconsistency in recording repeat results with the same person, or inter-observer, where there's a failure of different observers to record the same result. This often comes from training, but ultimately we're all human, and what we see is likely to be different from what somebody else sees. There are also technical limitations. The methods are unreliable or inaccurate, so this requires detailed validation before we can guarantee the performance of a particular assay. And system faults. Clearly, automation can go wrong. We've all suffered that in the past. Defective instruments, poor calibration, poor maintenance, lamps that go, and we're trying to ensure that the data that we generate tracks the performance of all the instrumentation on our deck, so that we can proactively and, more importantly, preemptively ensure maintenance. Now, if you look at the witch's broom of variability, as I've shown it at the bottom, I've introduced just 3 subjective intervention points. So these could be the addition of a reagent, these could be an analytical process, but ultimately each one requires subjective introduction of variability from a human. And the effect of this accumulation through multiple steps compromises discriminatory power. It results in the reporting of inaccurate data. It can adversely impact the quality of decision-making, and it most certainly increases costs and cycle times. And you can see that at each point from the reference value through to truth, with an error range of between 2 and 5%, every subjective intervention point could lead us further away from that true value. And this is something that is important to try and minimize. We appreciate that there will be a range of output, likely Gaussian distributed, depending upon the type of experiment we are doing, but ultimately it's keeping this range of potential outcomes as narrow as possible that is critical importance. So Hicham gave a very elegant introduction to the ACE2 spike biology, and just as a very brief recap in advance of a couple of data slides I wish to share. So SARS-CoV-2 infects the human respiratory epithelial cells through direct interaction between spike and a number of receptors. Obviously, ACE2 is the one that we are focused on as the topic of this webinar. The spike glycoprotein of coronaviruses contains protrusions which are essential for binding the virus to the host cell at the start of the infection and play a critical role in host specificity and viral infectivity. Spike is a large type 1 transmembrane protein. It contains 2 subunits, S1 and S2. The S1 subunit contains receptor binding domain, referred to as the RBD, which is responsible for recognizing the cell surface receptor, in this case, ACE2. And ACE2 and S2 contains the basic elements needed for membrane fusion. So the spike plays a key part in the induction of neutralizing antibody and T cell responses, as well as in protective immunity. So while Spike 2 as a protein ACE2 interaction is a key target for vaccine design, research is also exploring multiple modalities, including small molecules, neutralizing antibodies, and peptide therapeutics. Now, via mechanism 1, which is shown in the image on the left-hand side at the top, neutralizing antibodies could potentially block viral infection by binding the viral spike protein and preventing it from interacting with the cellular receptor ACE2. Mechanism 2, which is more on the internal side, neutralizing antibodies could potentially bind the viral spike protein and block conformational changes that the spike protein must undergo to facilitate fusion of the virus and host cell membrane. So there are a multitude of points in this cascade that one can intervene in. So what we're able to do at Arctoris is accelerate onboarding, which is the transition of validated assays from the external environment onto our deck. And what I'm referring to in classing it as assay onboarding is probably familiarly to you seen as the final stage in the assay development phase, where key assay parameters that have been defined are confirmed. An assay is transferred onto our automated platform and is encoded, ready for production screening. So the availability of high-quality validated reagents enables us to accelerate this onboarding, and we've done this across a number of assets in as little as 2 hours. More traditionally, it takes about a day. We use state-of-the-art dispense capabilities. Many of you will be familiar with the Dragonfly, the Tecan D300, and these enable independent reagent addition. They minimize dead volumes and ensure the highest level of assay performance, which is absolutely critical to the quality of data that we can deliver. What we were able to do here with the ACE2 Lumit assay is achieve successful onboarding within 1 day of receipt of just 200 data point 96 well pre-release kits. So this enabled us to generate all the data that we needed to confirm the validation and the performance of these assays. We were able to deliver robust signals of in excess of 20 to 1 with Z primes in excess of 0.8. We were able to show confirmed displacement using untagged recombinant control proteins, so both the ACE2 and Spike components of the interaction were put in as unlabeled and demonstrated displacement. We delivered near identical pharmacology with neutralizing antibodies, both positive and negative controls. And the 2 panels below here showcase the data that Hicham shared earlier and our own internal responses with the direct comparison of 3 antibodies obtained from Sinobiologics in China. Finally, we were able to validate a variety of molecular diversity. So the independent dispensing enabled us to generate high-resolution titrations, which can aid the characterization, in particular, of partial responses, which we see with a couple of the neutralizing antibodies. We've also been able to work with a client, a virtual biotech client, under contract to start assisting in their research. We have full displacement using unlabeled protein controls, where we validated the binding of labeled complex, returning the similar IC50s for both binding partners, as I alluded to. So the ACE2 is shown here on the left, and the spike interaction here is shown on the right. So really, to finish, the difference, this is a quote from Charles R. Swindoll, the difference between something good and something great is the attention to detail. And for us, every data point we generate matters. So in order for us to be able to deliver the highest quality data to our clients, we must work with high quality reagents, high quality automation, and really focus on ensuring that the details are properly thought through. And with that, I'd like to complete and hand over for questions.
Thank you, Dan. I'd like to now start the question and answer section of the webinar. To answer our questions, we've got Craig Malcolm from Promega, Daniel Thomas from Arctoris, and Lucy Wheatley from Promega. Lucy, do you have any questions to answer?
Yes, we've had a couple come in. Can you hear me okay?
Yeah, we can hear you.
Perfect. So the first question that came in during the Lumit presentation that Hicham gave was whether or not the antibody blockers have to be purified antibodies, or whether the cell supernatants or patient samples using serum can be used. And I think Hicham did go on to answer that, and the answer is yes, absolutely. We have done work with patient samples, and we can share that data with you. I think that has come in a couple of times actually. And we have another question here. How is the receptor binding domain produced, and what is the glycosylation state? That's a good question. That's something we'll have to look into for you. That's from Andrew. And so far, that's all the questions that we've got. So we can wait and see if anybody else has something they would like to answer. Okay, we have a question about using it in 384 well plate from John, and the answer is that I can tell you the volumes for the 96 well assay, which is that you use 5 microliters of sample, 10 microliters of your RBD reagent, 10 microliters of your ACE2 reagent, and then 25 microliters of the Lumit antibody mix. So that adds up to 50 microliters, and then the detection reagent is 12.5. And generally what we do is we would scale that down so long as you can keep the 2:2:1 ratio. So you would divide that by 4 to get your assay reagent for 384 well.
And to add to that, we have successfully done that. So our assays, all the data that we have shown is 384. We have successfully miniaturized it, maintaining those ratios, and that was what underpinned the Zs of 0.8 and above.
Thank you, Dan. And the question is, have we used the assay to test relative binding with the various spike mutants now circulating? I don't believe that's something that we've done, but it's certainly something that we can ask R&D whether we have any data or whether we intend to do it. That's a really good question. Does anybody else have anything else they'd like to ask? We will follow up with everybody who's asked questions to make sure that you get the data and the answers that you need. Okay, then I think I'll hand back to Hilary.
Thank you, Lucy, and thank you everyone for listening. We hope you've enjoyed that. If you do have any questions, like Lucy said, please do send them over to us. You can send them to me. My email's hilary.pollard@promega.com, or you can send, if you've got questions for Arctoris, you can send them to Dan. Thank you very much for listening, and see you soon.